So I'm delighted to introduce Dr. George Solomon from UAB, but who's going to talk to you about research. He has been instrumental in a number of clinical trials looking at therapeutic development. He runs a lab that looks at cells and how they function and how they are best perhaps integrated into using personalized medicine and trying to understand how medications, especially for rare mutations, might work. He's also the director of the adult program at UAB. So I'm really delighted that he can come and speak to us. Let me introduce Dr. Solomon, who's going to talk to you about what we've been doing in research and where we're going in the future.
So my presentations are always a little tech savvy, which means that sometimes they don't work. So I'm going to give you that up front. I was told last night when I had dinner with Peter and Noah and others that I was asked to come here because I'm an entertaining speaker, which means that not only do I have to explain some things about CF, but I also have to entertain you guys for an hour. So I'll do my best. I do have a funny accent. I'm from Alabama, so I grew up there. And I actually got involved with CF, and I'll tell you this story. Chad, you said that in about 2010, you were starting to hear about these modulators that might come around for you, or the future of those. And so in 2010, I was hearing about modulators from a very different angle. I had started in the middle of my internal medicine residency at UAB, not quite as vaunted a hospital as Hopkins is, but it's also a very good program, and we have a very big CF center there and have for some years. And so at that time, I elected to join a research residency. So I actually carved out some of my time during my second and third years of residency and did CF research clinic and was involved with a longitudinal research project, which was actually the exact same kind of research that Dr. Boyle was doing at that time, which is called nasal potential difference. And so we were tasked, we had a center there for standardization of that endpoint. And I got the illustrious task, because I had to learn to do the technique of making it better. So we could actually use it as what's called a proof of concept, which means that you take the mechanism of the drug from the lab into patients and show that it actually works. And so the concept was that one of the proof of concepts that these modulators were going to work, so we would test the drug working in cells that are responsive to those. And one of those areas is the nose, because the nose has epithelium or cells that line it just like the lungs does. So we could use that test. So that was how I got my input into CF, the CF world. And at that time, I thought, this is cool science. And at the end of that residency, I thought, these are even cooler patients. And so I wanted to do that. And so then I pursued my fellowship in Denver at National Jewish, where I did my fellowship for 4 years and did a research fellowship, and then was recruited back to UAB and have had a long career in CF now, or burgeoning career in CF, but the science started it, but the patients have continued it. So, and now my research is actually moving more towards patient research, and so I'm very happy that those two have melded so well, and many of us in CF have that same kind of story, that the science sparks us up front and the patients continue our interest. So your stories help us to continue that interest and renew us every day to stay up late and think about this disease over and over. So, all right, so let's see if I can make this work. So this talk, I have given a CF education day a couple other times at other centers, and most recently I gave it at the University of Minnesota. And so I've got some sciencey slides, and I've got some very non-sciencey slides trying to help us understand about how the gene works and all that. And I was going to make a Baltimore-focused non-sciencey slide that you guys will see in a little later, but as it turns out, the analogy was that there's a gate on a crab trap, and the crabs had to get out to rescue CF. But as it turns out, I love crabs so much, I did not want to rescue that gate. So I took those out, and I used the previous analogy, which was befitting for the Midwest, which is cows. And you guys may have heard that from previous plenaries. So I'll show those to you guys also to help maybe break it down even a little bit further so you can understand the science a bit more. So I'm just going to tell you that the crab prep analogy last night, I said, wait a minute, I don't want the crabs to get rescued. I want to eat them. So therefore, they're not going to come out. So, all right, so let's just move this over and we'll get started. So I've entitled this talk all about new CF therapies. Of course, I can't even begin to scratch all the surface, which is a great thing. But if I can't talk about something in an hour, it's wonderful because it means we have so much we're doing. So I'll hit the surface, and I'm going to try to, at minimum, get you guys to understand, because I have patients all the time that say, hey, I know I'm taking this drug, but I don't really know what in the heck it does, right? Or why it's going to help me and why you guys thought about doing it. So at minimum, we're going to talk about that. And then I hope that we will have a better understanding of some new therapies that are being developed and some new strategies that kind of line up with that Path to the Cure video. So I have some grant disclosures, and I didn't know what the policies were at Hopkins, so I put this slide up to be sure, but also it's a shameless plug to tell you guys about my 4-month-old baby. That's baby George. And so he was born very easily, and he's sporting his University of Alabama gear there, so roll tide if you guys are college football fans. I heard some Penn State earlier. Sorry, we don't like Penn State. We like them better than Ohio State, though. So, and better than Notre Dame. So this is George sporting his Bama gear a few weeks ago, and he's growing like a weed, and he's missing me today. So I'll be going home this afternoon to see him. So, okay, so the objectives are for this talk is to understand a little about how the CFTR gene and protein work, understand a little bit about how gene mutations cause disease in CF, understand what we know about modulators from the scientific side, not the patient side, and then what do we know about the effects of modulators? So what can we expect and what do we need to know to understand really what they're going to do for patients short and long term? And then there's aspects of CF care for patients that aren't covered by modulators and other elements of the disease that we're exploring in the CF world. So I'll give you guys a little bit of that flavor towards the end of the talk, okay? Okay, so I'm going to start with the survey. So by show of hands, who has had a family member or personally has participated in a research study? Okay, so a lot of people. Great, okay. Who has considered participating in a CF research study but hasn't yet? Family member, who's considered? Anybody considered but hasn't yet? Okay, so one hand, so maybe this will break down some barriers. It's not scary, so these guys are pretty nice. I've actually known the research coordinator team here at Hopkins for actually about the entire time I've been involved with research. I got to say they're one of the best, and they're very kind folks, so you guys should consider. And then who's currently taking a CF drug or has a family member taking a CF modulator drug? So a lot of people, and that number should get much higher as we go along and have this survey. You guys will have this survey in the future. So that's really good news. So that's great. So you guys are a really engaged group, and that's awesome. So hopefully you'll know some of this, and it'll be some review, or it'll be something new, and you'll have an understanding of what we're talking about. Okay, so we're going to start with thinking about how a gene causes disease. So this is the very far left. Is there a pointer by chance? Oh, so there's, oh, great, wonderful. This very far left graph is a general concept of how a genetic disease causes a problem. So in genetic diseases, genes are blueprints for making machines called proteins, okay? So you're talking about your son being an engineering person, and he's studying this. So the blueprint for a ride at Disney World is the gene, and the ride itself is the protein, and that does something to maintain health, some function in the body. And if the blueprint has a defect in it, the protein won't work to maintain health. And when proteins don't work, that's when you get disease in diseases like CF or genetic diseases, because this becomes a problem with this, and then the disease results because the protein won't work properly. In the case of CF, this is a sciencey diagram. The abnormal CF protein caused by the CF gene defect results in a bunch of problems with mucus, okay? And as a result, the mucus feeds people into this vicious cycle of recurrent obstructions of mucus and infections and inflammation, and that leads to a condition called bronchiectasis, which is dilation and damage of the airways, and that can cause end-stage lung disease. And this is what makes people have a lot of trouble in CF. Now, as you guys know, and as we've talked about a lot already today, there's more to the picture of CF because that protein doesn't just operate in the lungs. In fact, it operates in the sinuses, it operates in the liver, it operates in the pancreas in a big way, and that's what causes the plugging of ducts, so you don't secrete digestive enzymes. It works in the intestine to make little kids have meconium ileus because they get blockages, or it works for adults to cause obstructions or a lot of problems with gut function. And it also works in the reproductive tract to cause males with CF not to form the right tubules to be able to and have children. And so in the reproductive tract, it causes male infertility. And of course, as for a diagnostic purpose, it causes a skin defect, which makes the skin salty, as we mentioned earlier, and that's what led to the discovery of the sweat chloride test to measure that defect of the protein by measuring excess chloride anions on the skin surface, which is part of the salt. So this is looking down at the lungs. This is a video the CF Foundation made a few years ago. This will hopefully drive home what that mucus problem is doing in the lungs. So here we're zooming down on the cells that line the lungs, which have these things called cilia on them, these little red projections. And they sweep mucus and clear out stuff that the mucus traps, like infections and gunk that the lung inhales all day, okay? And that's regulated by this, which is the CF protein. The CF protein moves chloride ions, which are part of salt, and with that water, and regulates the mucus layer above these cilia to a happy state. So just like when you're doing your meditation, it's sort of a Zen state for the cells, right? So when the CF protein works, that's all great. But in the absence of where it doesn't work, and that regulation of that water can't happen, this is what happens. The mucus layer gets thicker, and it gets more difficult to move. And that paralyzes those cilia, which are little hairs to project and sweep that mucus out. And then the mucus gets really sticky, and it changes its properties, and then maybe you guys can see it gets full of infection, okay? And if without breaking that cycle, you have recurrent damage to your lungs, okay? So the aim of treatments with CF, this is the video in reverse now, is in fact to make the mucus move better, and maybe the cilia can move better. And until we had modulators or other drugs that worked on the basic defect, we were working on the mucus itself, basically, or components of it, like infection, okay? So what we hope to do is completely regulate the function of that CF protein, and that will better make the mucus clearance happen. So that's the real goal of all these therapies. And so how we get there tells us what level we're working on that process between gene, protein, and disease, okay? Are there questions at this point? You guys feel free to interrupt anytime we have questions. If we don't get through the whole talk, I'll just go longer, or we'll go after lunch. So, no, we won't go after lunch, I promise. So, okay, so here we're looking at that same pathologic process, but turned on its side, okay, and how it relates to therapies. So as we said, we have an abnormal gene. The abnormal gene is a blueprint for making a protein. So if the blueprint's wrong, the protein's going to be built wrong, okay? And then as a result of the abnormal protein, the protein not working or being abnormal, that alters the mucus properties and the ion transport, that chloride movement. It also makes inflammation and infection problematic, and then it leads to end-stage organ disease. And so what we're hoping to do is, and what we have accomplished at this point for many patients is, focus on the abnormal protein. So Cftr modulators, the reason they're called modulators is because they modulate or alter abnormal protein function. They do not work on the gene at all. The gene is still defective in patients that are on modulators, but the protein can be made to work better. Now, the reason that the genes weren't fixed first, which is, you might say, why not just start with this, is because that was the initial plan. The initial plan in the 1990s, once the CF gene was discovered, was fix this. But as it turns out, that is extremely hard to do. It's not easy. Many of us have been involved with trials, and only recently have there been some more movements towards that being more effective and having a sort of a curative type therapy. But as it turns out, there are ways to correct proteins. You can have molecules that bind to the protein, and they can do a job to make the protein actually work better without having to fix this kind of almighty barrier of fixing the gene directly, okay? Does that make sense to everyone? And to summarize, the key endpoints for developing modulators that were developed through the initial process of discovering ivacaftor and others was, let's improve lung function, because if we improve lung function, that's associated with better mortality and better morbidity for patients, and reduce exacerbation rate, also associated with improvement in mortality, and all the while improve symptoms for patients, both in the lungs and maybe outside the lungs also, because these drugs operate throughout the whole body. So that was kind of the blueprint for developing Cftr modulators. So for the next little while, I'm going to hopefully talk you through in a couple different analogies, one more sciencey, one less sciencey, how these modulators work and how the protein and drug and how the protein and gene interact together to make disease. Okay, so as we said, there's a lot of different genotypes in CF. The genotype tells us what type of defect is going to happen in the protein, okay? And what we've learned is, because Trikafta treats the most common genotype, which is F508del, okay, it treats patients with any of those, is the way to sort of think about genotypes in CF now is, do you have an F508 or not? And if you don't, then we need to think about different strategies. And so we're going to talk about some strategies for patients that have no copies of F508del a little bit later today. But we're going to focus first on what we know about treating F508del and what the kind of modulators are for that. And that's what got us scientifically to Trikafta and for other drugs that may help with people that aren't responsive to Trikafta in that space, or to augment our Trikafta's function. Okay, so this is a diagram showing the normal CFTR function. So here we're in a cell in the lung or nose or wherever else the CFTR protein's working in the body. Okay. In this case, it's a lung cell because we have cilia, where those sweep the mucus out, and we have a mucus layer. And in the normal CFTR function, what we see is that those anions, that chloride, moves through the protein as a channel. So in other words, the channel lets that anion move. And what that does is it regulates water. Okay. And when water is regulated on the cell surface, in other words, on the top of the cells where the mucus layer lies, okay, this keeps the mucus layer the right thickness, okay, and viscosity and stickiness, and that maintains health, okay? In the circumstance where you have CFTR functional defect because of the gene, the blueprint of the gene being defective, these different mutations prevent in different ways normal CF protein function. And they can do it by having no protein being made. That's the circumstance where patients that have an X mutation or a stop codon mutation, they have no protein being made. You can have where the CFTR protein is not stable at the surface of the cells where it's supposed to be. You can have where it's physically obstructed. That's the case of the G551D mutation. So the protein gets to where it's supposed to, but it can't open. It's obstructed from opening properly. Or the channel can't open in the right way, so there's no obstruction in the channel, but it can't open and close properly. So those are the type of mutations that are treated by kalydecoes we'll see in a few moments. And the loss of this function disrupts that normal anion transport. So the channel's supposed to be moving these anions like chloride and helping to hydrate the mucus layer and prevent thickening of the mucus layer, and then downstream problems like accumulation of mucus and paralysis of those cilia and whatnot, which cause problems in the lung and other organs. So the other organs are analogous. So in the pancreas, for instance, the ducts in the pancreas that secrete digestive enzymes, they get plugged with mucus and obstruction because of the same thing. They need the CFTR protein to help make that flow out of the tubules into the digestive tract. So it's the same kind of process, but we understand the best is what's happening in the lungs. Okay, so what we want to do is restore this movement of those anions back into that mucus layer and help with health. So, and what we think is that understanding how the CF protein functions and how the channel makes it to the cell surface will allow us to develop patient-specific modulators. So we've done this now, right? We now have patient-specific modulators, and the specificity is to the mutation, because the mutation tells us where the blueprint is defective, causing the protein not to be built properly, okay? So let's explore this a little bit. So there's a process here, okay? I'm going to talk through this quickly, in which the cells do a lot of different processes to make the gene become a protein and get it to where it's supposed to and do its job. So the first step is you have to have the DNA, the gene message, okay, operate to make a copy. So as you can imagine, if you have a hard drive on a computer with a copy of a blueprint, but you need to go out somewhere else and use that blueprint, you got to print out a copy of it, right? So the first thing this body does is it transcribes a copy of that into what's called a messenger or mRNA, okay? And the messenger and mRNA is the business working end of the blueprint. So it's a portable copy on your iPad or printed out on a piece of paper that the builder can use to make the protein properly, okay? But unfortunately, or fortunately for us for health, but unfortunately in CF, the body faithfully recapitulates a copy of the hard drive copy of the blueprint in the gene to the message. So the message tells exactly what the gene blueprint is supposed to do. So it doesn't correct it in that circumstance. It just makes a really, really good copy, okay? And so that means that then that message, right or wrong, if it's functional or not functional as a message to make a good protein, goes out and gets translated into a protein. So that's where the builder makes the protein out in this part of the body called the Golgi apparatus in a process called translation, okay? And after that, the protein gets checked, okay? So you've made the product along an assembly line. It's being built by the blueprint, and it either works or it doesn't work. And if it works, it goes to the cell surface and does its job, okay? And moves those anions, keeps the mucus happy, keeps the other organs happy, and you have health. If it doesn't work properly at that point, or if it's malfunctioning, the body says, nope, let's chew it up, and it chews it up what's called a proteasome, which is basically a part of the cells that chews up defunct proteins. And then, if it's working, the cell basically processes it and says, go ahead and do your job. So there's several different pathways on which the mutation can cause you to have defects of the protein to work or not work. And understanding these in even a simplistic way tells us why these modulators work differently. And it also helps us to understand a little bit, as we'll see in a few moments, why you have to have multiple modulators to treat defects like the F508del defect, which we're treating with Trikafta. Okay. So these mutations pair to the mutations affect different levels of this protein making machinery process. One of the more common of the less common mutations are these mutations called premature stop or X mutations. And they actually work by having a problem at the gene level, okay? So the gene level there makes a blueprint copy or a message RNA that says don't make protein, okay? And so therefore, these mutations are very severe because you don't have protein being made. In the circumstance of the F508del mutation or the G551D mutation, you do have protein being made. Remember we said that a modulator works on defective protein that's been made. So you can't modulate these types of mutations. And so we're going to talk about ways we're going to try to get around that a little bit later so we could make protein that could then be modulated, okay? And we actually have a process that we're doing that through our labs at UAB and the CF Foundation right now. And in this circumstance, you basically don't make enough, you make a message that says don't make protein. And so therefore, you don't go through this process of translating protein effectively and making it to the cell surface to do its job, okay? There are other mutations called splicing mutations that work very similarly. They basically make a message that's not effective. So the blueprint copy is not effective at being able to build a protein at all, okay? So they are somewhat analogous to the X or premature stop codon mutations. Now, let's think a bit about, this is where the technology sometimes doesn't work, so I apologize, guys. These slides were made by one of our grad students, and they're a little bit clunky at times, so I apologize depending on the computer. So now let's talk about the F508del mutation, okay? So it's a misfolding mutation. So in this, the blueprint copy is made perfectly, okay, except for it's missing one little part of the message to make a really perfect protein, okay? And what that causes is the protein machinery breaks down because it folds improperly on itself. What folding for proteins means is that it takes a strand of building blocks and they get assembled in a way that they can do their job as a machine, okay? So the building blocks then have to kind of fold themselves to make a machine, okay? And when that misfolding happens, the cell says, wait a minute, nope, chew it up and don't make it, okay? But what we've learned is that in addition to it being chewed up, some of that defective machinery, that misfolded machinery, in the case of the F508del, actually does get to the cell surface, okay? And it has a problem of its gating, or its opening and closing, just like the G551D mutation does. So that's why it took a drug that helps to correct the misfolding, okay? Like what's in our combi known as Lumacaftor, or what's in Symdeko known as Tezacaftor, or what's in tricaftor known as tezacaftor plus alexicaftor. That's the other we call corrector molecule. That helps with the misfolding. And then the third drug in those combinations is what? Does anybody know what that third drug is? What's the third drug? Kalydeco. It's because it works on the problem at the cell surface. So this mutation has a lot of different molecular defects. So that one little error in the blueprint results in a lot of errors in the protein to function as a machine. Okay? So we'll let the program run through itself. So in this, as we were saying, most of the protein that's being made goes through degradation because it's not supposed to be useful. Okay? But a little bit of it escapes the cell surface, and that can be rescued somewhat by the ivacaftor if we have enough rescue by those corrector molecules which make the protein sort of stabilize in its misfolded state. So it doesn't say go to that degradation method. Instead, let it assemble out beyond the factory line and go to where it's supposed to as a product. Okay? And then finally, mutations that reach the cell surface like the G551D mutation affect the opening and closing. And they have a more simplistic mechanism of defect. So they don't have a problem with making enough protein. They have a problem with the protein just having a functional problem. Okay? And so they get all the way through the translation and the post-translation, and they reach the cell surface, but they can't open properly. And so the G551D mutation was a common one that was discovered to have that problem, and it's very responsive to Kalydeco. And there are also other mutations called conductance mutations. A common one of those is R117H mutation, which some people have, about 5% of the patients have. And it causes a problem where the channels can open and close, but the pore doesn't have a nice, easy pathway for the movement of that water and anions to get through. And so therefore it's a conductance problem. The pore is not a nice, easy channel to get through. So it operates very similarly to the gating mutation in the sense that the movement is just impaired through, but the protein does get where it's supposed to. Okay. Now this is sciencey, so I want to pause and ask if anybody has any questions about how this operates. I'm hoping it's making some sense because this is the molecular understanding that if you can get this, you can understand why these modulators work because they're working on these problems. That's what these drugs are doing. So let's drill down on how the modulators work. So potentiators act on the protein where it's supposed to be, on the cell surface of the body's cells that line the organs, okay? And what it does is, like ivacaftor, okay, or Kalydeco, is it binds and bends the protein in a way that the protein just doesn't go back and shut. It just bends it open and so it can just let stuff flow. And so because of that, it works on mutations of both this conductance type and the gating type of defect, which are very similar. Now, and then allows the flow of anions, and then you have restoration of some health. Now, as we're aware, there are several combination drugs that include a drug called a corrector. Those include Orkambi, which we heard about earlier, Symdeco we heard about earlier, and then most recently, the addition of alexicaftor in combination with the two components of Symdeco to make tricaftor. Okay? And what those components do is they bind to the misfolded protein and cause it to operate better. Okay, and allow it to escape this degradation here in this degrading part of the cell and get to the cell surface, and then allow the ivacaftor to bind and do its job to bend the molecule, the protein open, with the addition of that molecule called ivacaftor and work. Okay, all right. So that's very sciencey. So now I'm going to get to our, and I don't know where my anyway. So I told you guys I was going to change these graphs over to crab traps, but I just couldn't do it because I love crabs too much, and I don't want to rescue any crabs, but cattle I do like. So I had developed these 2 years ago at one of the plenaries. One of the preeminent scientists in CFTR biology, Dr. Phil Smith, gave a discussion about, Phil Thomas gave a discussion and used this analogy about cattle being separated by a fence from the pasture they want to be to. So here we have cows, unhappy grass, no water. They'd like to be over in this pasture here where there's a nice stream for them to drink from and, a nice grass and alfalfa and everything that cows like. So this analogy worked very well for the Minnesotans because they have a lot of cows in the Midwest. So it may not work so well for folks in Baltimore, like I said, but I love crabs too much, so I could not change the analogy. So what we have here is a fence. And the fence is the separation of the cows, who are our chloride anions, okay, and the greener pasture, which is hydrating mucus, okay? That's the greener pasture in this analogy. But as you can tell, this fence has no way for those cows to get from this side, the nasty pasture, to the happy pasture, okay? So what we're saying here is just to remind ourselves that the message, the DNA, okay, is faulty. So the blueprint either doesn't allow any gates to be made, or when the gates get built into the fence, they don't work, okay? They don't work because there's not enough of them, or there's not enough of them and they don't open properly, okay? So that's the analogy here, just to try to make this simpler for you guys. Okay, so here's the circumstance where we have G551D. This is the gating mutation, right? So here we have plenty of gates in our fence separating our cows from the greener pasture, but the gates are locked shut, okay? So when we give patients with this G551D mutation, the potentiator Kalydeco, that acts like a key, okay? It opens the gate and happy cows go to the pasture, okay? So let's look at the circumstance where, this is where I put in my crab trap analogy. This is a happy crab because he's in the crab trap and he's about to be in my tummy here a little later when I get to the airport. So sorry I put that in the wrong place, I apologize. Here's the circumstance of F508del, okay? So this is the most common mutation, affects, depending on the population you survey, we oftentimes say 90% of patients. In some countries it's more like 80%, so somewhere between 80 and 90% of patients. And in this circumstance, remember I told you guys, there's a couple of different problems with F508del. The biggest problem is the blueprint causing a malfunctioning protein or a misfolded protein results in not much of it getting to the cell surface. So in this analogy, we don't have a lot of gates, okay? So we can give a corrector, as we talked about earlier, and the corrector can help us send a builder, add some gates to the fence, right? But the gates are still locked, okay? So because the gates are still locked, because the mutation doesn't cause it to work properly, we can then add that same key of Kalydeco or ivacaftor, then unlock the gates, happy cows back over in the greener pasture. Okay, now, this is going to kind of conclude the discussion about where we're at currently with modulators, because this gets us to Trikafta. Trikafta has two corrector molecules that operate at different areas of the protein to kind of synergize and make a lot of that misfolded protein get to where it's supposed to and allow the key or ivacaftor to work and open it, okay? So that's where we're at now, but as I told you guys, depending on the population, that doesn't affect everybody because some patients don't have an F508del mutation. So let's look at and review the history you talked about earlier in terms of patient numbers. So in 2012, there was the addition of ivacaftor for a few mutations, which affected about 10% of patients, okay? So that's those blue boxes here that happened in 2012 to 2015. Then we had the addition of Orkambi, okay, for patients that had 2 copies of the F508del. The corrector molecule in Orkambi was not effective enough to correct enough if you only had half of your genes being defective with the F508del. So it only worked on about an additional 40% of patients to help them. In 2019, we saw, of course, just about a little over a month ago, we saw the FDA press release and the approval of Trikafta, which now affects, as we saw here, an additional number of patients with a more effective modulator. So more effective here. You guys may hear this term as you're hearing more about modulators and other therapies for CF. More effective means that it has a response in lung function and other symptoms improvement on the order of what ivacaftor did. So we're talking about double-digit number of improvements in lung function and significant, highly significant improvements in symptoms and exacerbation rate. So what that means, by inference, is that Orkambi was not a highly effective modulator. It had some stability of lung function that went along with it and some reduction of symptoms and exacerbations, but not as good as Trikafta, we think, will, based on its effects. So this means that, depending on what numbers you use, 80 to 90% of patients now have a highly effective modulator if you have an F508del or you have a G551D or similar mutation. So that's where we're at. We were at the CF conference. We sang songs with Francis Collins and we had beautiful presentations, but then we were reminded, wait a minute, what in the heck? we've got a long way to go because there are hundreds of mutations in patients that are rarer that don't respond to any of these drugs, so they either don't make protein, or if they make protein, they don't get modulated by these current drugs that are available. So we've got to have a better way of dealing with that. And so what we don't know is when this year will be filled in this timeline. We hope it'll be soon. The reality is it may not be, though. And so what I'm going to talk about next in a few cases to hopefully stimulate you guys' interest is what I get asked because of the research that I do in our center, and our center gets asked to do, is asked by patients, how can we deal with this? And the second question I get asked very commonly, we'll talk about at the end, is what are we going to do about learning what this drug is going to do long-term for patients, like really answering the questions about the rest of the body, because we know that that drug is working on CFTR and other organs, but is it going to fix those organs or not? And will that be sustained? So that stimulated a little bit of my question to you earlier about will that be sustained for patients. We hope to answer that question. And finally, what are we going to do if patients have the onset of that vicious cycle of lung disease despite being on these modulators? What can we do about that to prevent that from getting worse while we're treating the CF mutation defect? Okay, so we'll talk about that for a few moments. Okay, so the next part of the talk is how we develop treatments and cures for all. And I'm not going to be able to answer this. I'm just going to be able to scratch the surface and talk a bit about the research that I'm helping to be involved with and the CF Foundation has led the charge on doing. And a lot of this will talk about some of the things that you guys saw summarized very nicely in that Path to the Cure video that Peter showed a few moments ago. So let's talk about some new concepts and review some cases. So the first case is AB. It's a parent of a 13-year-old patient with CF and F508del W1282X, and she says, well, I hear about these modulators, but you know what? I'd really like to have my family member have a gene replaced. Can you do that? And then can I have a therapy for my child in the meantime? So the answer 2 months ago was maybe. The answer now is yes for the second question, as you guys know, because what drug would you prescribe this patient now? Trikafta, exactly. Perfect patient for Trikafta, and it's actually approved for the 13-year-old now because it's approved for patients that are 12 and over, okay? So this is a yes, right? Right now, we can prescribe a drug for your family member. This is what it might do in the short term. This question is not a yes yet. And this question gets at, can we make a cure for everybody, and can it be adapted to be a one-time circumstance, okay? So we don't have to keep taking treatments and therapies and all that, okay? Okay, so let's go back to our cow analogy, okay? So remember that CFTR, the gene, causes a faulty message, okay? So the blueprint doesn't call for building the gates or the CFTR protein. What we can do with replacing the gene, though, is this would be a circumstance where we don't just fix the gate or build a little bit more gate, but we have the blueprint be fixed appropriately so the gate's inserted every time from the start and automatically opens and the cows are already doing what they're supposed to. So we don't have to make the disease better over time. We can make the disease never happen. That's the idea with a therapy that corrects the blueprint. And that's what's called gene therapy. Okay, genetic-based therapy, or the term that you may hear used some by us in the field is nucleic acid-based therapies, or it's therapies that work on the genetic component, not the protein component of the disease. Okay, that is what we hope to be able to do with the Path for Cure. Okay, so that's the concept there. So in that Path for Cure, I'm going to break down what's being done right now with this little flow diagram. So remember, we have a defective CF protein, CF gene. Okay. And there's a couple of ways we can work on that. We can fix the protein, which is what we're doing. And what we can do to expand the fixing the protein is we can actually deliver an effective message. So remember I told you guys that messenger RNA and that process called transcription is a hard copy for making protein. The gene itself, the hard drive copy of the blueprint doesn't really do anything except for makes a lot of these copies, the message copies, okay? So if we could deliver a healthy message copy, we might be able to make more protein in a way that would help people that don't make protein now, like the X or premature termination codon mutations. And then we also might be able to develop drugs that fix the protein problem by allowing that the translation. So remember I told you guys the translation is making the message into protein. And that doesn't work for a lot of the patients that don't currently have modulators available to them. So we might be able to fix these mutations by reading through the error in the blueprint copy or the message RNA, and that's called read-through. And I'll explain to you guys that in a few minutes with some diagrams that I hope will make some sense, okay? The other way to do it is, you know what, forget all this protein and message and all this business. That's too complicated. Let's just go back and make the gene therapy itself work. What's failed for years, let's actually just go ahead and correct the gene. There's two ways to think about doing that. One is just deliver the new gene. And so what has moved that science forward is there's been the development of to deliver a gene, you have to get it into the organ it's supposed to go to. And so there's been two major developments of doing that. One is tools that tell us that the cells are actually getting the right amount of DNA. Okay, that's one thing. Both functional tools and tools at the level of the cells themselves. Okay? The second has been moving the ball forward by making delivery systems that are safe. Because what we learned was that the initial delivery systems were developed were actually, you might take a guess as to what might be really good at getting stuff down into the lungs. Take a guess what that might be. What gets down to the lungs real well? Bacteria do. Think about another infection that causes a lot of problems people with CF besides bacteria. Colds. Viruses. Viruses are extremely good. And does anybody know how viruses actually cause disease? Does anybody know this? We learned this from the HIV epidemic some years ago. Viruses cause disease because they actually co-opt the cellular machinery to make proteins. They go in and insert their DNA into your DNA and make it take over, making more and more viruses. And when the viruses get made so many times in the cell lyses and it causes damage. That's why the flu virus is so damaging to people, even that are healthy, because it causes damage to the actual organ itself. And so the viruses were the first way of delivering these genes down to the lung and other organs. Well, as it turns out, people got sick with viruses. And it caused a lot of inflammation. So we had to back up. The world of scientists that study that had to back up and say, let's develop new mains that aren't viruses or that are safer viruses to deliver the genes down into the lungs and the organs. And so that's what's being done now. So those are the two things that have moved this better forward. And we think we have a better way moving forward to get that to happen. Now, the other thing we can do is fix the gene. So what if you could go in and just cut out the bad part of the hard drive copy of the mutation, right, and put in the right part? And as it turns out, a few years ago, there was a development of a system that can do that called CRISPR-Cas9. Has anyone ever heard of that before? Have heard of that. Great. So CRISPR-Cas9 is what's called a gene editing technique. There are others. That's the one that's the best known. So I'm just introducing it. There are others that do that also. And actually, the CRISPR-Cas9 system is being developed through an NIH study to treat sickle cell anemia. Anybody familiar with sickle cell anemia in here? So that's actually being studied, and the initial results have been very positive at the NIH for that. So they're actually editing the gene that causes sickle cell anemia and having dramatic effects. So that could be a strategy beyond replacing the gene. You could go and edit the gene. If you ever edited the gene once correctly and efficiently, you're home free, right? Because it's just fixed forever, right? It's built into the computer hard drive. So it always has the right messages and always makes the right proteins at that point. So those are the strategies that we're looking at. So let's look a little bit more at what's been done and what's been known in CF looking at these type of therapies. So this is a science diagram from an mRNA replacement therapy, okay, that actually we deliver to the lungs, and it works to actually repair the mRNA itself. Okay, so the message gets fixed by this drug called alufracin. This is a study that we helped to lead at UAB, and what this study shows is this is looking at an endpoint called nasal potential difference, is that in this study, when you give alufracin to patients that have a particular defective protein or defective message, their movement of anions on using that drug actually gets better. So this arrow shows patients that had the drug versus those that got placebo. And when this line goes down, it means you're restoring health. So what this told us was, in this proof of concept study I told you guys about earlier, is that this drug actually works to edit the defective message, the message RNA, and makes you make protein. And when the protein is edited in this way, it works at the cell surface to actually move those anions and create some restoration of health. So this is a study we published year or two ago. We're proud this is going to move forward into more later term phase trials to look at repairing the message of the defective gene. Okay, now here's another example. So this is a study from the UK. They have what's called the Gene Therapy Consortium in Britain. And it's been led by Eric Alton and some other scientists for years trying to develop gene-based therapies. And this is, once they got this non-viral vector figured out, I told you guys about earlier. So an inert particle encapsulates the DNA and allows it to insert into the lungs. Then they said, will this work? Okay, and so the first question they asked was, if we give this down into the lungs, we nebulize that inert particle containing the DNA inside it, will it get into the cells? And what this tells us, this shows us here is that, in fact, this group of patients here that got the drug had their cells lining the lungs, had, in fact, incorporated the DNA into them, okay? Whereas those that got placebo had no incorporation of normal DNA, okay? And by a similar functional assay, so they actually stuck a bronchoscope down in the patient's lungs and measured the movement of anions directly in the lung, okay, a procedure we also do it in Birmingham, and they found that, in fact, compared to the placebo, which was having no effect on the chloride movement, these patients had restoration of the chloride movement with that. So what I'm showing you guys this to say is that we're already making strides and proving this concept can work, and now it's just a matter of honing the trials the right way so we can actually test it in people for more effective means of doing this. And this is just a couple of samples of what's been done. There are other examples that you would find in the medical literature or you may hear about, but these are a couple of examples that I have the most experience with. So what this tells us is that the gene correction, editing, that kind of concept is viable, and it can be used for patients agnostic. In other words, independent of what their actual mutation is, because you can deliver this gene to anybody with CF, regardless of what their mutations are, okay? So we're making paths on that cure. So that video is not just some sort of, highbrow, lame brain, we're going to make this at some point. We're actually doing this, right? And so this is what's great about it. The CF Foundation's now put the money oomph behind it to make sure it happens in a more accelerated fashion rather than just slower process. So, okay, now, let's turn to our next patient. So this is a 25-year-old female person with CF, presents to your clinic for treatment recommendations. And she has this genotype, W1282X, G542X. She's a high sweat chloride, and she has several questions of you. Am I eligible for triple combination therapy? What's the answer to that? The answer is no, unfortunately not. Why is that, though? Scientifically, why is that? Who wants to tell me why that's the case? From what we've learned today. Yeah, does it make protein? Exactly. Modulators require that you make protein. Stop codons, like the X mutations, don't make much protein to modulate. So we have to figure out ways to either fix the gene or fix the message in a way that we can make protein and then we can modulate it, we think. Okay? Does that make sense to everyone, that concept? Okay? So the step we're missing, and that is making enough protein to modulate, to fix that for patients in the short term and have a treatment. Independent of having a cure for that, a treatment at least. Okay? So, what kind of mutation or X mutations? We talked about this briefly. They're stop codons. What they do is they create faulty messages that have a message inside them called a stop codon. What a stop codon is, it says, stop making protein, exhaust your efforts on the factory line. This message is telling you to make a piece of a protein. That piece of a protein will not do what it's supposed to. Okay? So, therefore, you get very little or no protein being made that can do its job, okay? And our next question is, what strategies are being developed to treat this unique type of my CFTR defect? I've always been told I have a rare thing, it's a more challenging thing. What can I do to fix this? Okay, so one of the things that's being looked at is small molecules to actually read through that stop. So I'm going to endeavor in my best efforts to show you guys how to read through agents, because those have been in development for some years and likely will reach patients before we hope. I'm not going to say likely. We hope we'll reach patients before we've worked out all this gene therapy stuff, because that's going to take some time, okay? So this is one of our sort of shorter-term shots on goal, is create a drug that would read through the stop message and make some protein we could then modulate, okay? So that's what's called a read-through agent. And I'll tell you guys, The read-through concept actually was developed from a drug that commonly treats CF patients now. Does anybody know what that is? Know what the original read-through agents were? Concept? It's an antibiotic, actually. The antibiotic is the aminoglycoside antibiotics, and one of those is called amikacin or tobramycin, okay? Who in here's heard of tobramycin before? Probably everybody, right? So those drugs actually work on bacteria, and they poison the bacteria's ribosomes. Ribosomes are what makes proteins. So it was discovered that maybe we could modify that function and make us poison ribosomes that make stop codons happen in human cells, okay? So that's where the concept came out. And the initial studies that read through were actually giving high doses of aminoglycoside antibiotics to patients to see if that would promote read-through. And the concept sort of worked, but didn't work perfectly. And then there was a study of a drug called adalurin. Some of you might be familiar with that, which didn't cause read-through enough in CF patients, but has actually been approved in Europe for other diseases where there's X mutations, like, I'm trying to remember the type of drug diseases those are. Duchenne's muscular dystrophy. Those are diseases that cause neuromuscular defects in kids. Very, very debilitating illnesses. So they actually work for that, and they've been approved for that purpose in those types of diseases. So, but it didn't work in CF. There was a large number of studies, and those were conducted about the same time initially as the ivacaftor studies were, but they just didn't kind of reach the endpoint we wanted to. So we're in the process now of thinking about new ways to develop read-through agents. So let's talk about that for a moment. Okay, so remember, we're going to revisit the premature termination or stop codon mutations, and they're unstable, and they make no functional CFTR protein, or really no protein, even non-functional for that matter, because the message is defective totally there. So let's zoom in on that process, that transcription process we talked about earlier, which is the process of taking a message, the printed out copy of the blueprint, and making the machine the protein, okay? So in the normal circumstance, okay, transcription moves the message from the cell nucleus. You make a copy, a good, faithful copy of the gene, and make that message and send it out to the cell machinery that makes proteins, that assembles proteins. And I just introduced that to you guys. It's called a ribosome, and they're a part of this endoplasmic reticulum which lines the nucleus. It's what we need to know. But the ribosomes there assemble the building blocks of proteins there called amino acids, one by one, okay, until they hit a stop message, which says, wait a minute, we've made all the protein, the full-length protein, stop efforts, send it off the assembly line, we'll do the rest of the processing some other place in the factory, okay? So that's the normal process, okay? And in that system, what happens is the ribosome, this little oval here, reads down the message, makes a protein, that's the yellow ribbon here, and you have normal CFTR protein doing its job, and it goes to the regular processes and goes out to the cell surface. Now, in the circumstance of the stop codons, the transcript is abnormal, and the transcript abnormality is, like I told you guys earlier, it has a stop early message. It has a stop short message, what that means. So you're not just stopping early in number, you're stopping short in ever making the protein to work at all, okay? So here's what happens. So here's the example of W1282X. It's one of the more common of these X mutations. So you try to make a message, you're printing out a copy of it. Here's the little guy, the photocopy stand, he's printing out a copy of it. But the message has got a part that's not going to make any protein. That's that orange part right there. You guys see that? Keep that in mind for the next couple of diagrams, okay? So that process doesn't quite get through, so the message is faithful, but the faithful message is not functional, okay? So then that goes out to its normal part of the body. Let's zoom in again how we're making this protein. So in this circumstance, the message for making protein has a premature stop in it, so it's going to stop the translation or the making of protein from message. That looks like this. There's the stop right there at the part of that orange part of the message. So short protein is made, and it doesn't work. It's too short to do the early stop, okay? Therefore, it doesn't work. Now, let's say we add a drug that causes read-through. Here's our read-through agent. Still have a stop mutation, right? Add a read-through agent. Happy drug, right? Protein gets made fully. Now you have normal functioning proteins you can modulate, okay? So that's what a read-through agent does. It disrupts the assembly of a ribosome that brings in a stop, okay? So it says, I don't care if there's a stop there, ignore it. Put an amino acid there anyway. Put a building block there anyway. Even if it's imperfect, we'll work through that with the modulator. Put it there anyway. Make a full-length protein. Then we can use these drugs to make the protein work, okay? So developing these read-through agents, that sounds easy, right? Well, it's not, as it turns out. It's extremely challenging because doing this for CF, you have to restore a lot of CF protein function to actually restore health. A restoration of a small amount does not work for CF. You have to restore a lot, especially in the lungs. So unfortunately, what we have is we have a lot of compounds that restore a little bit of read-through. Okay, they read through that a little bit, but not enough to reach patients, enough to affect endpoints like improving lung function. And that's what happened with atalurin. The atalurin drug works brilliantly. It does cause read-through in cells, but it doesn't do it enough to make it have an endpoint for patients that you can say, all right, this is really working for them. So what we're doing now is we're trying to develop means to test these compounds, okay, and we're studying 2 huge libraries of drugs that might work for read-through that have been sort of developed for other purposes, and we're testing those with a bunch of assays that have an endpoint. The endpoint is, did we make a read-through message or not? Okay, so suffice to say that our labs in both the CF Foundation and UAB and some other centers are testing almost 1 million drugs. You heard me right, almost 1 million drugs that might do this to determine if that's going to work or not. And if it does, then the next step would be test in patients with markers of whether or not it's working or not, and then go to just trials for these rare mutations, okay? So the last case is going to get at what we're doing to develop markers for patients that would tell us if a rare mutation might respond to a drug now or in the future, okay? So this is the case of EF, 21-year-old white female person with CF, has 2 rare mutations, okay, D1152H, and this rare here, there's actually a splice mutation, okay? She's got a sweat chloride that's in the 60s because of this D1152H actually has some partial function, so it actually has some mutation, some protein that works as a result of that mutation, but her lung function's in the 60s, so obviously she wants therapies to prevent lung function declines. And so she asks, am I eligible for approved modulators? And your answer to her will be what? What's that?
Maybe.
Yes, because of D1152H. Well, maybe. So it's a question. That's a question. So so she's kind of got a maybe, but at present we don't know. And so that maybe begs the question, well, could we test if she's going to respond and figure that out? Because she's got, how many patients are really going to have that combination of mutations to study? So how could we work that? So that's exactly, the maybe is an important part of that. So it's no or a maybe, right? Is there a strategy for developing modulators or other therapies for these types of patients? The answer is yes. Okay, so the CF Foundation, in their always infinite brilliance, came to us and said, we want to develop a biorepository of human tissue, okay, to do 2 things. One is study patients that have rare mutations, the first of which is X mutations, so patients that have 2 copies of X mutations. That's about 2 to 3% of patients with CF have 2 copies of X mutations. And the other is patients like our third case here that have mutations that might respond to a modulator, might not, might get benefit, might not, but there are not enough of any of those to study in a large-scale clinical trial. Okay, so the idea was, can we get tissue from lots of different parts of the body, develop markers that tell us, 1, does the drug work in the lab, in the test tube, and 2, will that predict how the patients are going to do, so we can modify our routine clinical trial design from having these big, huge, randomized clinical trials to smaller trials with some prediction based on what the test tube told us, okay? So we can move the drugs forward faster. So that was the impetus for this rare study. And the rare study is capturing patients with multiple different rare mutation combinations to do this kind of work, okay, in a couple of steps. The step now is get a bunch of tissue, see how the tissue works. The next step is develop clinical trials down the road, okay? So what we're doing with this study is 2 things. As I told you guys, we're taking tissue from lots of different parts of the body, the lungs, way up here in the nose, that's cut off up here on the person, the intestines, because the intestines actually can make endpoint called organoids, which I'll show you guys about in a few moments. And then we take the cells, biobank them, we study them in different aspects of our labs for how they culture, what they do as far as expressing gene and protein, and then what do they do? So if we coax them into some sort of differentiation state, will they do something like we saw with those functional assays with the gene therapy? Can we, in the test tube, can we let them show they're working, they're having something that makes them show the function of the protein is working or not working? And then we hope eventually to correlate this to clinical improvements observed with these drugs and see if we can tell, all right, this mutation, in fact, would respond to a modulator because in the test tube, we give that modulator to this particular patient with this rare mutations, and they get this response. And that's similar to the response that patients with F508 got for Cftr, right, for instance. Does that make sense? So we correlate to what we know about other more common modulators and more common mutations and see what happens, okay? So what have we done? So the first thing we did was we looked at, there was data out of the Netherlands in the development of these things called organoids, okay? And what organoids are is, as opposed to making a line of cells cultured up and having them do something that shows that the CF protein's working or not working, you make a ball of them like this, okay? And the ball operates as it can deflate. You guys remember from Deflategate with Tom Brady, right? So it can be deflated and then you throw the ball easier, right? But the ball doesn't work as well, right? Or you can have it, that's more point for you, yeah. So, or you can have the ball inflated up. And what inflates a little ball with organoids is what? Water. From what? You know this. You know this. What inflates it? The water inflates it by what? Because the CFTR protein works, and it makes the chloride move in, and the water follows, right? And so if they inflate, that means the protein is working, okay? So here's what we're showing. So this is, I told you guys, one of the things we want to do is develop read-through agents and use these biomarkers to test them in people, right, for these X mutation patients. So this is videos of a patient with this mutation, G542X, R553X, two X mutations that make no protein. And this is when we treated the organoids from the patient's bottom. Okay, I'm not going to say the other word, bottom. And they, in fact, do nothing. The cells don't swell when we give them Orkambi, essentially. However, when we give drugs which are being developed through this process, what we see is these are read-through agents. One of these is a toolkit reagent we can't give to patients. This one might be able to be given to patients. And we also give them Orkambi. What happens? These little things swell. And they're swelling because the CFTR is being activated. Now, the nice thing about this endpoint is those folks in the Netherlands have already done the clinical correlation to lung function and sweat chloride for us. So if we can hone this assay for developing in rare mutations like the X mutations, we'll have a way forward to be able to predict clinical response for patients and maybe move read-through agents forward and test them in a quicker fashion. Now, I'm saying this is a maybe. This is not a definite. It's just the first step on this pathway to potentially doing this, okay? So then we have to know, well, how do we conduct trials for patients that have really ultra-rare mutations? So the concept that we've been developing, and this has also been developed at Cincinnati Children's Hospital and lots of other places, is N-of-1 trials. What an N-of-1 trial is, as opposed to having lots of people and you take a group response in an endpoint like lung function and see if it improves the drug, here you predict the patient will have a response and you say it definitively on the one patient through repeated testing. And if that works, then you give them drug, okay? So this is a whole different kind of clinical trial design, not one that's currently endorsed by the FDA, but we hope one day might be for other diseases as well as CF. And so the concept here is, the first step is you have a biomarker that you've shown really definitively tells us whether the patient's likely to respond to the drug or not. So it might be the organoids, right? You give your organoids this drug, it has this response at a cutoff, and that means probably going to have a lot of restoration of CFTR protein function, right? Then you do on-off studies of the drug and see what happens in those periods. So you have drug treatment on and off, on and off, on and off for a long period of time. This is a clinical trial design of we call our theratyping study at UAB. And then we measure a lot of things that tell us if it's safe, and then is it activating CFTR? That's what all these boxes are telling us. And does it improve lung function, other endpoints like symptoms and whatnot, like we see with the other modulators. So this is a concept of how we can develop a trial for more personalized towards patients that have rare mutations that we can't study in larger clinical settings, okay? All right, now, last case. We're on the home stretch. You guys have been really patient with me today, so we're on the home stretch. So this is the case of GH, 29-year-old person with CF. She has F508del homozygous Chloride or combi. She's about to switch over to Trikafta. And she says, I have the following questions about Trikafta for you, doctor. What will be the long-term effects on my lungs? Can I expect long-term effects on infection and nutrition? Can I come off my enzymes? How will my diabetes happen? Can I stop my pulmozyme, my hypersal? I've got so many questions, and I want to know the answers right now. And of course, the answer to all this is we don't know, but we're going to find out, okay? So here's how we're going to find out, okay? So the one thing we're going to find out by is by the PROMISE study. So I actually had a discussion with these guys yesterday because they're going to be one of the sites for this. UAB is one of the lead sites for the PROMISE study. And the PROMISE study is, the concept is that after the drug's approved, we study patients at the time they're getting their drug, before they've gotten the drug effects, and then monitor for years in an observational study after for lots of different questions. Okay, so here's the study design. Not important for you guys, except for know that we're going to enroll 400 patients of a mix of patients that have 1 or 2 copies of F508del, and they'll have lots of visits over the course of years. Okay, this study will probably be extended beyond just those 6 visits, which will happen over the course of about 18 months. Probably we hope to 3 or 4 or 5 years to answer longer term questions. Okay, and what the questions are is this. So this is going back way back to that slide I showed you guys an hour ago about disease manifestation. And we've got sub-studies in every one of these organ systems hoping to look at what happens in the lungs. Do the liver disease, do you unpack the tubules in the liver by restoration of CFTR function? Do we restore the secretion of digestive enzymes? And do we help the intestinal health so that patients have normal motility of their gut and don't get DOs? We won't answer the question of reproductive tract because we think that's embryologically programmed and that can't be fixed. But we will look at the sinuses in a sub-study at UAB looking at does the sinus inflammation and mucus movement there improve also. So the idea of this study is we don't just answer a question of short term we have a therapy that unpacks mucus and restores lung function in the short term like we know from the published studies about Trikafta. We want to know is this going to be long term, and will giving a drug that activates CFTR throughout the whole body like it does, will that have effects similarly in the other organ systems beyond just the lungs? And so that's the idea. So we don't have data on this yet. We know from similar studies with ivacaftor called the GOLD study that some of these get better and persist. We don't know all of this. And so we've developed a lot more sophisticated endpoints, especially in the liver, pancreas, and intestine world, so we can really answer these questions. And answer a question like, here's the cutoff of restoration of enzyme function whereby you can stop your enzymes or you can't stop your enzymes. Or you can do this or you can expect this with your intestinal health. So these are the kind of questions we're going to try to answer with this study and others. Now there's other things that are being done with modulators. The Simplify study was mentioned earlier. I'm not one of the lead persons, but I have been involved a little bit with the discussions about this. The idea here is the most common question I get with Trikafta is, well, can I just stop? I hate taking hypersal. Like I've never met anybody that really liked taking hypersal. Maybe somebody does, but none of my patients do. Can I stop taking hypersal? Can I stop pulmozyme? Can I go to more infrequent dosing with these? I mean, the mucus is better now. We don't know, and so the Simplify study is going to look at that in a unique trial design of withdrawal and determine if we withdraw, do we have preservation of health or do we have what we call non-inferiority? In other words, is the drug as good as taking the drug plus the other drugs like pulmozyme and hypertonic saline with lung health? So that study will be conducted, probably enrolling, were you talking about the study? Probably what, mid next year, I think is the spring, mid next year. So look out, you guys will probably be a site for it, I would guess. So look out if you're interested in that. Very interesting trial design. I was fascinated by the statistical analysis that went by to design the study. So it's very interesting. So, and the next thing is there's other modulators being developed. Okay, so there's a company called Proteostasis. They have a novel modulator design called an enhancer and an amplifier. And the amplifier works to actually make more of the message. Okay, so if you make more message, what's going to happen to the amount of protein being made, do you think? Increase, right? So the idea here is that you bump up the number of messages, you can make more protein, and then let the correctors and potentiators work on more protein being made per unit of time. Okay, so The idea eventually is we hope to pair the amplifier, if it works, with the Trikafta or their combination to see if we can have even better results for patients with F5-Whitney, especially with more severe disease and whatnot. So, and then there's a study going on now for once daily ivacaftor being led at multiple centers where we could take out that afternoon dosing with the Trikafta or patients that are staying on just ivacaftor therapy. And then there's also Trikafta being studied in new mutations that we didn't think were possible in the test tube to see if they might be able to extend the label of Trikafta to some additional mutations down the road. So that's being done by Vertex right now. Okay, so the last thing is, I remember this was the blueprint for how a gene becomes disease from earlier, and I told you guys that what happens at the end result of this is that there's a lot of a vicious cycle of infection, mucus obstruction, and inflammation, and that results in end-stage lung disease. And so though we think modulators and cure for all will affect in some patients, like kind of nix this out, it won't for everyone, right? There's still in patients with advanced disease going to have infection issues, mucus obstruction, inflammation, so we have to keep targeting that. So the CF Foundation went all in last year on a huge amount of money to study these aspects of disease and health in CF. And so I'm going to tell you guys a little bit about the studies that are ongoing for that. So again, we're going to focus back on that pathologic process and treatments for that. And here we're going to emphasize mucus abnormalities and inflammation in the lungs. There's several studies being looked at. One of those is studies investigating the proper antibiotic administration for exacerbations. There's the STOP-2 study, which is an adult. It's undergoing right now. You guys are a big center for that. In fact, Natalie West is one of the lead PIs for that here. And then there's a STOP-PED study. Is it started yet? I can't remember. It's starting next year. I don't do PEDs, so I'm not familiar with it. Starting next year. So the idea here is people still will get exacerbations. How do we best treat those so we affect the best outcomes for patients? There's also studies looking at treatments for MRSA, which is a gaining ground as a common pathogen, especially in advanced lung disease, and mycobacterial disease. Anybody familiar with MAC or sepsis? So those are gaining ground as people age with CF, as more common infections and can cause a lot of problems. And so there are a lot of therapies looking at ways to target that, both therapeutically and observational, to understand how the disease and how that infection interacts with the disease. We're also looking at new approaches to Pseudomonas, including new methods of treating the infection beyond routine antibiotics, and then also looking at approaches to treat and resolve chronic inflammation. Because one thing we have learned, and it's a huge unanswered question, is even with highly effective modulators, there's no definitive evidence that the inflammation in the lung actually totally resolves like you might think. And so that means there may still be issues with ongoing damage of the airways that we want to see if we can make better. So there are definitely a lot of studies, especially looking at the blocking of an enzyme called neutrophil elastase, which is a common pathogen, a common protein that causes damage to the lungs in response to infections and inflammation. So this is a little sampling of what's happening in this world. And so with that, I'm going to conclude, and thank you guys for your attention and your interactiveness. And so I hope this made some sense to you guys and it brought to light some of the questions, and I'll be happy to take any questions you may have about any of this science. So. We've only gone over 20 minutes. Okay, be happy to take any questions you guys have. Yes, Vera. Oh, sorry. We got to wait for the mic. The webcast. Sorry about.
Thanks, Marty. That was really a great talk. You made it really easy to understand, so thank you. My question, though, is we hear a lot about like one-time cure, and we kind of group in those mRNA therapies and gene therapy. Can you describe what's the difference between those and what is actually a one-time cure?
Okay, so that's a great question. So one-time cure would require a permanent either editing or incorporation of the normal genetic material into the cells of target, right? Say lung, for instance, okay? The current strategies being developed are not one-time cures. They are delivery of some material. So the mRNA-based therapies will require recurrent nebulizations or administrations because mRNA is inherently not very stable to do that. So it gets used and it gets degraded intentionally, okay, because it's supposed to be a message not to make too much. So that one, any mRNA therapy would have to be recycled and was given over and over. Some genetic therapies, like the editing techniques, have been shown in preclinical models to be somewhat permanent. So if you edit that out and you get it going in the right process, it can become permanent. Now, what we don't know is a few things. The first is, when do you have to administer that in life to be permanent with editing? In other words, will the body revert or something like, or figure out what you've edited, right? And then how much editing efficiency has to happen to be permanent or sort of permanent. Does that make sense what I'm saying? So there's a lot of unanswered questions, but the only thing that would be a permanent cure would be, let me give you the perfect scenario for a permanent cure. 100% efficacy of a systemically administered editing or gene therapy. Okay, so you give it in the bloodstream, it goes to every body system and 100% efficiently gives it, and it never goes away. The body never thinks through it because it's so efficient. That will take a long time. We probably will make incremental steps in that, and that's what's been done. The cancer world has done a lot of this kind of work, and they've been incremental steps in that, not fix, right? That will probably take a long time for a permanent fix. So, does that answer your question? Okay. Other questions you guys have? Yeah.
I've been reading about phage therapies
Yep.
for resistant bacteria. Can you speak to that?
Absolutely, sure. I'm sorry I didn't mention that. So, phage therapy, there's a couple labs moving that forward. The group at Yale has been pretty aggressive with that, and there's some others that are doing that work as well. Phage is actually a primordial virus that affects bacteria. And they are, for whatever reason, they're there, and so we can co-opt this concept of a phage to poison bacteria we don't want to have there. So the concept here is multi-drug resistant Pseudomonas, is that we could give a phage that targets your particular strain of Pseudomonas and basically goes in there and selectively kills the Pseudomonas in a way that bacteria, that antibiotics can't do. And so basically it's working the same way we talk about viruses incorporating into cells to give DNA. Here we're using them as a strategy to safely kill selectively bacteria, but not our human cells, basically. Does that answer your question?
Yes, it does.
Yeah.
Are there currently ongoing studies?
There are. There's a study at Yale, but it's only at their center. We actually were thinking about being a site for it, but there've been some. Without getting too far into the regulatory world, it's not being done under kind of the standard new drug application process, but rather a waivered new drug application. So it's a little bit of a regulatory hurdle to get over to do that. So they're doing it at their center. So I know they would be willing to take consultation, but then we're not doing it at other sites yet. So, okay, other questions? Those are good questions.
Probably have time for one more if there is.
One
or-
more? Yeah, one more question?
Mm-hmm.
Or I can meet with you guys at lunch.
If
So-
not, we'll thank Dr. Solomon and